The very time you are talking about cell cycle, you just don't say that you have this one cell which is dividing into two cells. You get it? This one cell is dividing into two cells. If you are just saying that this is cell cycle, no, this is not cell cycle. Cell is not, it is not just cell division, you are saying. You get it, right? It is not just cell division, but cell division comprises of 5% of total cell cycle. Cell is not only dividing, but cell is preparing itself. You get it, right? The cell will prepare itself to divide, and then the cell will divide. Are you getting my words? Right? Cell will have to do a lot of things. It will have to duplicate its DNA, it will have to duplicate all of the cell organelles it is having in its body. It will be requiring some of the proteins for spindle fiber formation. It will have to increase in the size of cell growth. And then when everything would take place, at that particular time, after that the cell will divide. Get it? So cell division is just a subset of cell cycle. And cell cycle comprises of all those activities where a cell prepares itself to divide and finally it divides. You get it? When you talk about cell cycle, so the on time, see, I'll make your cycle like this. Say for 95% of the cell cycle is belonging to preparation activities. Correct? It is all about the preparation activities. How? Say I divide this something like, for this complete period, the cell sit properly. Why will you wait? So listen here. So for this particular period, say more than 95% of period, the cell is preparing itself, and that particular complete period is known as interphase. So interphase is that particular period which comprises of more than 95% of cell cycle, where a cell is what? Where a cell is preparing itself to divide. Got it? Which again comprises of three substages. The interphase is that particular timing where the cell is preparing itself to divide, and that is again divided into three substages. These substages are G1, US, and G2. Get it? Got it? And in say less than 5% of cell cycle, this divides, which is known as M phase. Got it? So in G1 as G2, the cell will be preparing itself, and in M phase, the cell will be dividing into two cells. Are you getting my point? Yes? So this G denotes gap. This is also a gap. Got it? Gap is to prepare something. Gap is to fulfill all the requirements. Those are needed. There are two important processes going on. One is cell division, and one is this S is called synthesis, and this is also known as synthetic phase. So some synthesis is going over here. Are you getting it? Some synthesis is going in this particular phase. What synthesis? The DNA synthesis is going on. Because when a cell will have to divide into two cells, cell will have to duplicate its DNA so that it can send one copy of DNA to one cell and another copy of DNA to another cell. So cell will have to duplicate its DNA. Did you get it? So the DNA should be duplicated over here in what phase? S phase. And DNA replication would be requiring some enzymes. You have heard in class 12 you must have read about DNA replication. So DNA replication, it requires many enzymes, right? So all those enzymes like gyrase, topoisomerase, SSB proteins, all those enzymes are formed in G1. So proteins related to what? Proteins or enzymes, those are required for what? For DNA replication. Should form where? Should form in G1. G1 is gap, and this gap will fulfill the requirements those are needed in us. Got it? something which will be required in M phase, that particular requirement would be fulfilled in G2. You got it? So what would happen? All the proteins those are required for DNA replication would synthesize here because DNA synthesis is going to happen here. Is it all okay? Now apart from protein synthesis, most of your cell organelles, the cell organelle duplication also takes place here. The cell organelles double here. You getting it right? The cell will be looking for doubling of all those organelles because it will have to send all those organelles to different cells. So most of the cell organelles get doubled here, except 3 organelles. Those get doubled at G2. There are 3 different organelles. These 3 different organelles are chloroplast, Golgi body, and mitochondria. Those will get doubled over here in G2. And rest, all other cell organelles are getting doubled over here in G1. Is that all okay? Yes. Apart from proteins required for replication, the cell also requires a RNA primer. You heard about in DNA replication you require an RNA primer. That RNA primer is also formed in G1 because why? Because DNA replication is going to happen over here. For this DNA replication, you require this RNA primer. So the RNA primer is forming in G1. Got it? But rest all RNA forms from where? From DNA. No? This DNA to RNA formation, you call it as what? Transcription. So DNA would be forming RNA. Once the DNA would form, and the DNA is getting formed here, it means all other RNA would form here in G2. So all 3 types of RNA, mRNA, tRNA, rRNA are formed in what? G2. So all other type of RNA are formed in this particular phase. Got it? The primer RNA which is required for DNA replication would form in G1, but rest of RNA would form in this particular phase because the DNA is forming over here. Is that all okay? Yes? Proteins, RNA, cell organelles, everything is going over here. So in most of the time in G1, the cell increases in its size. The size of the cell would increase in G1. S phase is looking for DNA synthesis. The DNA, extra copy of DNA is forming here in S phase. Not only DNA is getting duplicated, but something else is getting duplicated, which we call it as centriole. Now the centriole is required for esterase formation. This will be going to opposite poles and will be helping the spindle fiber arrangements. Getting? So the centriole should also duplicate. You are okay with it? Centriole will be duplicating in S phase, and one more thing you should keep it in mind, that centriole is never present in plants. You are okay with it? Centriole is never present in plants, so it is this particular step is only happening in where? Animals. See what would happen? Where is this DNA synthesis going on? This is your cell. This is your nucleus. The DNA is present where? In the nucleus. The DNA to DNA formation, where did I tell you? Where will it take place? In the nucleus itself. So the nucleus DNA to DNA is forming. That means DNA duplication. Whatever is going in S phase, it's going inside the nucleus. So in the DNA to DNA formation, the nucleus is forming that particular DNA. Is that fine for you? Now centriole is present in the cytoplasm. DNA is present in the nucleus. Centriole is present in what? Cytoplasm. So centriole duplication takes place where? In cytoplasm. And one centriole will start moving to one pole, another centriole will come to opposite pole, and then the spindle fiber would form. DNA would form the chromosome, and spindle and chromosome joining, so that what should go? The nuclear membrane should disappear in prophase, which would be happening somewhere in time. Did you get something? Are you getting it right? But what you call the centrioles are never formed in plant cell. That doesn't mean the plant cell will never be forming spindles. You get it? Centrioles will just provide the space to spindle to get arranged. But centrioles are never forming spindle. What is forming spindle is a protein called tubulin. And tubulin forms in plants as well as in animals. Get it? And tubulin is required for what? Spindle fiber formation. The spindle fiber formation will be taking place where? In the cell division part. So spindle fiber formation requires what? Tubulin protein. So that protein should form where? In the G2 itself. So in G2, one more protein called tubulin forms. Is that all okay? You getting it right? Now, This S phase, apart from DNA synthesis, apart from central duplication, will also look for stone protein formation. The stone proteins would form here in what? S. Why? What are they required for? Histone. What are they required actually for? They are required to combine with DNA because DNA is acidic and it is negative charged. Histone proteins are rich in basic amino acids. Yes. So when they are rich in basic amino acids, they could what? They could combine with DNA. So you have the DNA copy forming. You should require histone instantly, so that what would form? The chromatid would form somewhere. And this chromatid would be jumping here in M to form the chromosome, and that's how the chromosome would be moving and duplicating, and you will see the cell cycle complete. Is that fine for you? समझ में आ रहा है कुछ? कुछ कभी कोई doubt है तो you ask me. Last boy, any problem here? Am I watching to you? Where's your friend? Other friend? Where did he go? He was there, right? He was there before. Where did he go? You don't know. So see you. Now what happens? This is all okay? Is that fine for you? Right. Now what happens? Let's suppose that a cell, listen to me, a cell has 2C amount of DNA here in G1. 2C is the amount of DNA. 2C means, let's suppose the cell is having 10 picogram. Now picogram is the unit where it's a unit to put, it's a picogram, it's a small unit, right? So that is basically unit what you represent the DNA amount in. So let's suppose a cell is having 10 picogram of DNA. After S phase, how much picogram of DNA should be there in G2? 7, because DNA synthesis is going on, right? So what would be the amount of DNA here? If you have 10 picogram, it would be what? 20 picogram here? Got it? But if the cell is having 46 chromosomes here, there's a difference between DNA and chromosome. The DNA forms chromosome, that's all. But here, there are no chromosomes. The chromosome would be forming in M phase, right? If there are 46 chromosomes here in G1, there would be only 46 chromosomes here. The chromosome number would not get double because the chromosomes are never forming here, the chromosomes would be forming here. Is that fine for you? So most of the time they would ask you, if the amount of DNA is this here, let's say X here, and the amount of chromosome is Y, you get it? Let's suppose the amount of DNA is X and the number of chromosome is Y, what would be the amount of DNA and number of chromosomes here in G2? So amount of DNA would be 2X and number of chromosomes would be Y only. Is that fine? Later on, this particular M phase where the cell actually divides is been divided into, I've taught you this particular process, right? Where a cell will have to divide, first of all, what will it do? It would divide its nucleus, right? It would look for, how will it go? It would first divide into its nucleus, and then it would be dividing into what? Two cells. So first of all, division of one nucleus is what? Karyokinesis, and this is what? Cytokinesis. So in M phase also, there are two things. One is karyokinesis, another is cytokinesis. So you can divide into two parts, karyokinesis and cytokinesis. Is that fine? This is your karyokinesis, and this is cytokinesis. Now karyokinesis is again subdivided into four substages. These are prophase, metaphase, anaphase, telophase, and furthermore the cytokinesis comes. So karyokinesis is divided into these four stages, and then you will get the cytokinesis. It means the cell will be forming two cells here. Got it? The one cell which is jumping into G1 will be forming two cells here after cytokinesis. You get it? Now sometimes the questions are asked, listen, the questions will be asked, let me give you some options. Say G1, S, M, G2, G1, G2, M, S, G2, M, G2, M, G1, S, and the fourth option, this is third, fourth option is say S, G2, M, say G1, and then M. You have to answer me, what is the correct sequence of cell cycle? What is the correct sequence of cell cycle? You get it? No. When I'm asking you the correct sequence of cell cycle, the cell cycle should follow in this fashion. It doesn't mean the cell is always in G1. None of your cells are like, it's not like all of your cells are in G1 now. All the cells will start at the same time in G1, no. Some of the cells are in this place, some of the cells are in this place. But when the cell is undergoing the cell cycle, it will have to follow a fashion. Just after G1 S comes, okay, but just after S, the M doesn't come. G1, no G2. But this is M. G2, M, G1, and S. So this is the correct answer. Most of the students look for, it should start with G1, no. It should follow the sequence. It can start from anywhere. Got my points? Now, furthermore, what happens? This is all clear to you? Any other problem? Any problem here? You getting it? Now what happens? Say there is a cell which is dividing into two cells. Two cells will again jump into G1, would form you 4 cells. They would again jump in G1, would form you 8 cells. But let's suppose some of the cells are not willing to go into further cell cycle. They don't want to go into cell cycle further. Got it? So those cells will sit into G1 only. You get it? Will sit into G1 only and will never go in what? Cell cycle. They would sit into the sub-stage of G1, which we call it as C0 stage or quiescent stage. In quiescent stage, the cells are active, but they are not proliferating. Proliferation means division. They are not dividing. They can divide if you put some drug and all. If you call them to divide, then they will divide. Are you getting it? Like root meristem you have. Remember? Root meristem, just after the apex, root apex, you have the root meristem. In meristem, the cells are dividing continuously. But in meristematic region, there is a center called quiescent center where the cells don't divide. Got it? That particular center is called quiescent center where the cells will never divide. And these cells will only be dividing when the root would be getting the wounds. Are you getting it? So quiescent center cells can divide when they are in need of. Is that all okay? Now cells most of the time they sit in C0 stage, that is why they are so long. Now cells you see, right? They are so long. Just because of that the cells are not dividing. When they are not dividing, they are what? Continuously elongating. And that's what now cells are for elongating cells. Got it? Now, G1 can be called as post-mitotic. Post-mitotic, after mitosis is this happening. And this stage we can call it as pre-mitotic stage. Tell me. Yes or no? Pre-mitotic is just before mitosis and just after mitosis. So post-mitotic and pre-mitotic stages. Is that all okay? Now let me tell you something else.
Sir,
Yes.
battery is so low.
Shut up.